So today we're going to talk about Koch postulates for the microbiology course for Dr. Cotter. Now this is going to be on your first knowledge of celebration, and also you're going to see this type of material covered on the final as well. So during the finals week, you should probably rewatch this video because you probably forgot a couple of the steps that you're going to probably need to know for your final. Now, the Coach Postulates has 4 steps. The first step is going to be examine the patient for signs and symptoms of an illness. Then you're going to want to collect samples from that patient to be able to do further testing in the future steps. So, a sign is what you as a healthcare worker will be able to observe or measure on a patient. So, you could be observing bad breath when you're looking at someone's throat. If they have a oral infection or throat infection, you may be able to notice some bad breath. You may also see some inflammation, so that's going to be some swelliness and some redness at the site of the inflammation. Also, if you're checking someone's temperature, you may notice a fever because the body temperature is elevated, and you can measure that by looking at a thermometer, and it's elevated compared to a normal range for a person. You can also check to see if the person has a high blood pressure or a low blood pressure. That's something you can measure as well. Now, something you can observe is maybe a skin rash or a lesion or abscess on the skin. Now, a symptom is what a patient is experiencing that you, as a healthcare worker, will not be able to measure or observe. So, you're going to want to ask closed or open-ended questions. So, you can ask them, "Where is your pain located?" And they could say, "Oh, it's my pain's located at the throat or chest or on my right hand or arm, or it's in my stomach that I have some pain." Then you can ask, "What type of pain do you have? Is it a dull, sharp, aching pain?" And they can be able to let you know what type of pain that they're going through. Now you could ask them, "What else are you feeling other than the pain?" And they could say, "Well, I'm very fatigued lately. I'm feeling dizzy or nauseous." Now, these are other symptoms that you cannot be able to see from a patient, but this is what a patient can report to you. So a symptom is something that a patient's experiencing that you can't observe. And a sign is something that you can observe and measure on a patient. So once you figure out the signs and symptoms of the illness that you're working with, you're going to want to collect samples from that patient. So these could be a urine sample, a blood sample, a fecal sample, or you could do a biopsy where you cut part of the infected part to be able to look through a microscope, or you can use a swab to be able to swab like a throat to see if it's strep throat as well. Now, this leads to step two. Step two, you're going to want to isolate the disease-causing microbe by using some lab tests. So what we're going to want to do is we're going to want to grow, or what we like to use in our lab, term culture, and we're going to try to identify those microbes by using those lab tests. We're going to be covering these lab tests in lecture and lab to be able to know what tests each test tests for to be able to identify the microbes. So you may see on your knowledge of celebration, please identify 2 or 3 laboratory tests that you are going to run for to be able to identify the microbe that your patient has that's causing the disease or illness. And what are they testing for? So a microscopic test will be looking for the cell size and shape. Also, you have a macroscopic test where you're going to be able to look at the colony size and the shape, and that's going to be this part here. This is looking at the colony size with the dilutions, which we're going to be doing in a further lab down the road. We also have a chemical test to identify the ribosome subunits. So if it's a prozonan or a fungal infection, it's going to have a ribosomal unit of 6040, and a bacterial sub ribosomal subunit is going to be 5030. We're going to discuss this a little bit later in further chapters, but the chemical test is going to identify the ribosomal subunits, And then with that, we're going to be able to identify if it's going to be a bacterial or if it's going to be maybe a protozoan or a fungal and causing disease. Now we have also a metabolic test, and this is going to see if the microbe is a fermenter. And then the other test we have too that we can run is a genetic test. And each microbe has their own DNA sequence. So once we identify the DNA sequence, we can look through the database to figure out which microbe it is because each microbe has its own DNA sequence. And we can go through the system and isolate which microbe it is due to the DNA sequence that we identified. Also, you can do a blood test to be able to find the antibodies that are located in there to also try to figure out what the body's fighting um, off. Now, step three, what we're going to want to do is once we isolated that microbe that we believe is the disease-causing microbe, we're going to want to infect a new host. It could be an animal or a healthy cell. And what we want to do is we want to observe the same signs and symptoms from the original patient in step one. So we are going to want to look for the same signs and the same symptoms that the that we noticed as healthcare workers from our patient and what the patient reported um, to us as well. That could be the inflammation, bad breath, would be the signs, and then the symptoms could be the pain was located in my throat and it was a um, dull sensation of pain, and I feel very dizzy and I was very fatigued that the patient may report that you're going to want to see in the new healthy host that was infected. Now, step four, we're going to want to isolate that same causing disease in our new host that we infected in step three. And we're going to want to do the same steps in step two, where we will grow the slash culture the microbe and run laboratory tests that we performed in the second step to be able to identify the microbe. So we're going to probably want to run the same test. So if we did a blood test in step two, we're going to definitely want to run a blood test again. And if we used a genetic test as well, we're going to want to do that again for step four. So make sure you use the same laboratory test that you used in step two to be able to identify the same microbe that is causing the disease or illness from our original patient and our new healthy patient that we infected in step three.